Hello, this week we are starting to learn about photosynthesis, the other kind of major metabolic pathway in living things. Today we're going to start with just a real brief introduction to photosynthesis, and then we're going to be going into the lab that you picked up some equipment for last week. So first of all, photosynthesis is a process you have learned before. Can you write the chemical equation for this process? Hopefully you wrote down something kind of like this. If you did not write the balanced equation with the sixes, that's okay. But hopefully you came up with the fact that photosynthesis requires carbon dioxide and water and energy from sunlight. And it produces carbohydrates like glucose and oxygen. One little caveat with this is how we write the equation for this reaction. The process of photosynthesis doesn't directly make glucose. That's actually kind of a step beyond the actual reaction of photosynthesis. It makes a 3-carbon sugar that's kind of a precursor to glucose. In general, you can think of this as kind of a simplified equation. For every carbon dioxide and water molecule, we make just a generic carbohydrate. This isn't an actual sugar. It's just a generic equation for a carbohydrate. Carbohydrates always have twice as many hydrogens as they have carbon and oxygen is released as a waste product. So really, this is a process of building carbohydrates 1 carbon at a time, and then they just keep linking these molecules together to make sugars like glucose. That's a 6-carbon sugar. Photosynthesis is another example of a redox reaction. Remember, as we talk about all of these metabolic pathways, we're really looking at how energy is transferred on electrons that usually travel with the hydrogen. So if we look at this reaction, the hydrogen is here at the beginning, and it has those high energy electrons. And in the end, it's over here, and there's even more of those electrons. So the hydrogen got transferred from here, and when this loses its hydrogen, it becomes oxygen. So that's kind of the water is what's donating the electrons. So in the language of redox reactions, this is being oxidized. I'm not going to write the whole word because that would take forever. The carbon dioxide is what eventually gains those hydrogens to become this glucose molecule. So it is what is being reduced in this reaction to become that carbohydrate molecule. So photosynthesis is a reaction that's building up molecules. So this is an anabolic reaction that requires an input of energy that comes from sunlight. If we look at just an overview of kind of how this process happens, we'll be looking at this in more detail on Thursday this week, but you can see the inputs. It requires sunlight, water, and carbon dioxide, and there are kind of 2 main steps that both happen within the chloroplast of a plant cell or the cell of another organism that can do photosynthesis. The first step is called the light reactions. It takes water and the energy from sunlight. It produces a little bit of ATP and this molecule NADPH. This is another one of those electron carriers. And oxygen is kind of a waste product of that first step. The ATP is then used to take carbon dioxide and the hydrogen that's brought over on that electron carrier and use that to build glucose molecules, to build those sugars in a process known as the Calvin cycle. So those are kind of the 2 main steps, and we will go more in detail with those on Thursday. Today, though, I just want you to think a little bit about how this process of photosynthesis is connected to cellular respiration and what factors might affect the rate or the speed at which photosynthesis happens. So you're going to be studying both of these processes by looking at an organism that can do both. Remember, last week we said that every single living thing needs to do some form of cellular respiration to make ATP. So organisms that also photosynthesize, things like plants and algae, can do both photosynthesis and cellular respiration. So you took home a lab kit last week that looks like this. One of the items that you have in your lab kit is a little vial. This is called a cuvette that has a bunch of little green beads in it. Those beads contain algae that are able to do both photosynthesis and cellular respiration. And we will talk a little more as you start getting into the pre-lab and looking at how this works. You'll think a little more about when they will carry out the process of photosynthesis and when they would carry out the process of cellular respiration. And you're going to be kind of studying both of those using this same system. So the lab that you're going to perform, this is actually going to be your task for tomorrow because the lab itself takes about 45 minutes, and it also requires a little bit of setup. One thing that I do want to point out here, you can see across the bottom of the screen kind of the color chart, which you have also in your lab packet. You brought this home as well. You may notice on yours, the very last color over here, the way it printed, it looks dark blue. As you're measuring your results in this lab, you might want to even write a note on there that that final color is actually supposed to be a very dark purple. You will not see a blue like it printed on your sheet. It should be a very nice dark purple. The lab instructions are posted in the homework section. There's a whole lab document here, and you're going to be working through a bunch of background information and some pre-lab questions here in just a minute. But once you get to about, I don't know where I am, the third page or so, you'll see the actual procedure from the lab. One thing that's really important is that your algae beads need a little bit of time to kind of activate and wake up before this lab will really work. So before you do your experiment, you will need to take these out of the refrigerator and put them somewhere that is well lit. So this little container with your algae beads needs to come out of the refrigerator and sit somewhere with some good bright light. It doesn't need to be super close to a lamp or anything, but you just want it out where there is some light for about 4 hours. The best place to put it would be sitting in a very sunny window. But otherwise, just a nice brightly lit room or underneath a lamp on your desk or something like that is fine. That will just help these algae kind of wake up and begin photosynthesizing a little bit because they haven't been doing that so much when they were sitting in your dark refrigerator. When you're ready to actually do your experiment, you have a few other things in your kit here as well. So you will take the cover of these little cuvettes comes off. Be careful not to spill everything out of there. And then you're just going to use your pipette to suck out all of the water that's in here, and you can just discard it. I do not have a beaker sitting next to me, so I'm not going to discard mine right now. But you just want to suck it out until all of the water is gone. You can reach it way down into the corner and suck out every little bit of water from those algae beads. So all you have left in here are the little green algae beads themselves. Then you also have a container right here of carbon dioxide indicator solution in your little yellow vial. You can pop the cover open on there, and you are going to put 1 milliliter of this carbon dioxide solution into the cuvette with your algae beads. You can measure 1 milliliter using your pipette. The mark that is right up here near the top, you'll see a little line, is 1 milliliter. So you want to fill it to there. If it's a little above, that's okay. And put that much of your carbon dioxide indicator back into the cuvette with your beads. And then you get to decide where you are going to test. I'm going to run a couple experiments tomorrow where one of my cuvettes will be sitting under a nice bright plant growing light, and another one will be kept completely in the dark. You get to kind of set up a third experimental trial. So you can think about a different type of light that you could use. You could think about a different color of light if you have something that you can use to kind of change the color of light at home. You can think about changing the temperature or the distance away from the light or any of those factors. But make sure you keep track of what you're doing and record what your setup looks like, because we're going to be discussing that on Friday. Once you've decided where you're going to put it, make sure when you set your cuvette under the light that the clear side, if you look at these, some of the sides, two of the sides are really clear and the other sides have some lines. Those lines will actually block some of the light. So make sure one of the clear sides is up, and also make sure that all of your algae beads are laying flat across the bottom. They're not stacked up on top of each other. So just kind of shake it around until they're all laying flat so they get as much light as possible. When you set it in your experimental condition, you'll start your timer. And then every 5 minutes, you're going to compare the color of the indicator in your cuvette with your color guide right here, keeping in mind that this very last color here should be a nice dark purple instead of a blue. And record the pH as closely as you can in a data table. We will be looking at this data again on Friday. So let me just pull this back up and move myself out of the way. So tomorrow you will be doing a few things. First of all, today you are going to work through the pre-lab questions. So that's your first task as soon as you finish watching this, is to go and begin working through the pre-lab questions to make sure you understand how this indicator works and when it's going to turn more purple and when it's going to turn more yellow, and what your data means and kind of what's going on in there. Tomorrow is going to be a little bit different. Tomorrow is not a normal Wednesday. We're actually supposed to have live class sessions tomorrow, but because we're doing this lab, the live session will be optional. Your main assignment for tomorrow, and this will be posted in Classroom tomorrow, but your main task tomorrow is to perform this experiment. There will be an optional live session if you want to come in and ask any questions or work on the questions, the pre-lab questions with some group members. You can do that. You're going to perform your experiment at home and collect your data. And also in the packet that that we worked on earlier in this unit, you may have noticed on the page with the mitochondria, there's also a diagram of a chloroplast. The other thing I would like you to do before Thursday's class is to label the parts of your chloroplast, color them if you choose, and make sure you're familiar with that vocabulary before Thursday. If you have any questions as you begin working through the pre-lab today, you can let me know. You can work on everything up through about question 11 and start brainstorming how you're going to run your experiment so that you are ready to do that and collect your data tomorrow. Let me know if you have any questions, and I will see you in a couple days.